Ben, and thanks for the invitation to speak. I just started my lab last July, so most of my talk will be about work that I did as a postdoc in Matthew Meyerson's lab at Dana-Farber in the Broad, along with work with the collaborator Catherine Wu. So here's a figure from one of the descriptions of the pan-cancer analysis projects. And the goal of the project was to look at various omic types, such as mutation, copy number, gene expression, as a way to look at all the ways that cancer genomes are altered, look across multiple tumor types, and try to identify the different pathways that are altered. And so something that's missing from this figure is the contributions from alternative splicing. And so the main interest of my group is to look at ways that splicing is altered in cancer transcriptomes and to understand how that's actually contributing to tumor genesis. So briefly, what is splicing and alternative splicing? So essentially every gene in the human genome is alternatively spliced. Genes start out for the pre-mRNA, and you can either decide to include or skip a portion of that pre-mRNA. Typically, we describe them as, say, exons, so you can have either exon inclusion or exon skipping. And these different decisions can actually have a functional effect on the gene. So this is just one example of the FAS gene that can have inclusion or skipping of exon 6, and this has very different functions to the protein. One's a membrane-bound receptor, one is soluble, and they have very antagonistic functions. And so one indication that splicing was really important in cancer was that recent whole exome sequencing studies had found that many recurrent mutations occurred in splicing factors. So these typically were in proteins that were part of the core spliceosome. So here I'm showing two examples of those splicing factors that have been found to be mutated, which I'll describe in the talk today. So one is U2AF1, and this is part of the U2AF complex that's involved in 3' splice site recognition. And another factor is called SF3B1, which associates with the branch point sequence within an intron and is also thought to direct splicing. So what's very striking about many of these splicing factor mutations is that they occur across many tumor types, both in solid tumors and blood malignancies. The big question in the field is to understand how these mutations are contributing to tumor genesis. So why are these mutations being selected for? And one kind of common hypothesis is that these mutations, since they're splicing factors, are affecting the transcriptome. And so if we were able to then look at the set of altered exon splicing events, potentially you may expect retained introns or unsplicing of introns. You may also observe gene expression changes due to these mutations. And if we were able to then identify the collection of all these transcriptome alterations, we could then potentially look at what are the affected genes or pathways. So when I started my postdoc, I looked into the Cancer Genome Atlas, which was a great data source because for each tumor sample as a part of the TCGA, we had both whole exome sequencing and matched mRNA sequencing from the same tumor. And so we were able to then look for splicing alterations in the same tumors that had these mutations. So the first gene that we looked at was U2AF1. So what I'm showing are just hotspot mutations in U2AF1. There were additional mutations found across other portions of the protein, but this S34 position is the most frequently mutated position in U2AF1. And what we also saw by looking across all the TCGA tumors was that this U2AF1 gene was most frequently mutated in AML and lung adenocarcinoma, and so those were the two tumor types that we followed up on. So I'm simplifying a lot, but essentially what we did was to look for splicing changes due to this mutation. We took the collection of patients that showed no mutations in any splicing factor and compared them with the set of patients that had mutations in this splicing factor. Took the RNA-seq data from those patients and looked for differential splicing. So the approach that we used to look for differential splicing was to use JuncBase. So this was a program I originally developed in my graduate work that I've since been updating for improvement with cancer genome studies. So the main benefit of JuncBase is that it's able to detect splicing events directly from the RNA-seq data without the need for an annotation. And actually, this is really important when thinking about looking for splicing changes due to a splicing factor mutation. We may expect that many splicing changes have never been seen before, since these are mutations in core factors. And so with JuncBase, you're able to detect alternative splicing. It also classifies the splicing in these different types, which I'll show is also very informative to study these factors. And JuncBase can also quantify alternative splicing. And I'll go through one example of that. So this is a representation of an alternative 3' splice site event, where in the white box is representing an alternative exon or a portion of an exon. And there's two different isoform choices. There's one that includes the white exon, and there's one that skips the white exon. What's represented in red are RNA-seq reads that would support inclusion of that isoform. And in blue, I'm showing representation of reads that would support skipping. And so JuncBase will identify these reads and assign the reads to either the inclusion or exclusion isoform. From those reads, it calculates an abundance of the isoform, and then of the two different isoforms. And then from that, it can calculate a % spliced in, which is essentially the proportion of the inclusion isoform that's expressed relative to the total abundance. And so for this one PSI value, we can quantify this PSI value for every patient sample and for every splicing event, and then we can perform differential splicing analysis on this value. So here I'm showing a real plot of PSI values for one particular splicing event, and what we're doing is we're comparing the PSI values between wild type and mutated samples. And so we can do this for every event, and then after multiple testing correction, we can get a set of strongly associated splicing changes that are correlating with this mutation. So for lung adenocarcinoma and AML, we looked into the splicing changes that were associated with this mutation. And what I'm showing is what classification of those splicing events that we saw. And so we saw primarily cassette exon type events being affected, as well as alternative 3' splice site events that were affected. And this was actually promising because this was what we expected, given the known function of U2AF1 as part of the U2AF complex that's involved in 3' splice site recognition. And I apologize, it's really small, but we saw a total of 131 splicing changes in lung adenocarcinoma and 369 strongly associated splicing changes in AML. So these mutations seem to be affecting hundreds and hundreds of splicing changes. And so one of the challenges, and something that I'm planning to continue in my own lab, is to try to understand what the functional consequences of these splicing changes are. And so here I'm just showing 3 genes that were shown to be affected, given some of their known roles in cancer, as well as RNA processing. So one we were particularly interested in was beta-catenin, as that's a gene that's frequently mutated in cancers, primarily through missense mutation. But this is just to illustrate some of the challenges of understanding the consequence of these mutations. So beta-catenin, the event we saw was altered in both AML and lung adenocarcinoma. And we could show that this was actually caused by the mutation by these validation experiments where we expressed wild type and mutated forms of the protein, and could show that indeed the mutation does cause a splicing alteration of beta-catenin. So this event occurred in the 3' UTR of beta-catenin. We tried to follow up a little bit on if it's affecting beta-catenin expression or downstream pathways, or the pathway that beta-catenin is involved in, and we didn't see anything obvious from that analysis. So it's still unclear, even though we're seeing this very conserved splicing change, it's unclear how that's actually affecting the pathway. So what I just described was our studies on the effects of U2F1 mutation. And so another way that we could perhaps understand the functions of these mutations is to look at other splicing factors and maybe compare what are those transcriptome effects. So another one that we looked at is SF3B1. So again, it's a factor that's associated with the branch point and is also involved in regulation of splicing. So SF3B1 is actually one of the most prevalent mutations in chronic lymphocytic leukemia. And so this work is in collaboration with Catherine Wu's lab. So this is a review from the Wu lab showing the locations of the most frequent SF3B1 mutations in CLL. And so these do occur in this heat repeat domain. And the most common, the most frequently mutated position is this K700E. So again, we can use the same approach. So take the samples that have no mutations in splicing factors and compare those with the samples that have the mutation and look for splicing differences. So again, we see that SF3B1 mutation causes altered 3' splice site usage. However, unlike U2F1, we don't see as many cassette exon cases, which here is shown light blue. So SF3B1 seems to strongly affect 3' splice site changes, as well as differential intron usage. We've done some additional functional studies of some of these splicing changes to understand what's functionally happening with these mutations. One kind of computational look is just to ask how many of these mutations are in frame and out of frame. So in the presence of the mutation, we're seeing a splicing switch, and that would be in the majority of cases predicted to be an out of frame mutation. So would thus be predicted to be a loss of function for those genes. Another thing to point out is that many of these splicing changes involved usage of a novel splice site that was never annotated. So again, it's really important in these studies that when we're looking at splicing factor mutations to look at potentially unannotated splicing changes. So one of the interesting in-frame deletions, so just to go back, so most things were out of frame, but we were also interested in genes that might fall into this category where they're in frame. That may indicate that the splicing change is potentially gain of function or a change of function in that gene. And so one of the genes we have followed up with is DVL2 or disheveled. So the Wnt pathway is well known to be altered in CLL, and disheveled is this component of the pathway. And what I'm showing here is RNA-seq coverage along this event. So the alternative 3' splice site is here, and down below is showing one transcript annotation. And so what you can see is that in the presence of the mutation, a more downstream 3' splice site is being used. So you can see the drop in coverage in the mutated sample. And again, not an annotated splice site. And so some functional follow-up that my collaborator, Kathy Wu and Lily Wang have done is to show that this splicing event actually does have a functional effect on the protein. So just to summarize kind of what we're seeing with these splicing factor mutations and how we're sort of framing it is that these mutations, like the known transcription factor mutations, are having very pleiotropic effects. So classically P53, one of the most frequently mutated genes in cancer, is known to affect many, many different downstream pathways and have many different effects on the cell. And so we can kind of think of it similar with splicing factors. These are global gene regulators. And what we're seeing based on our transcriptome analyses is that we see many different pathways that are directly being affected by these differences. And another interesting point is that based on our studies and other studies on these splicing factors, as well as others that I haven't shown, it does appear that these mutations have different effects on the cell. So there's different sets of splicing and gene expression changes. Sometimes you do see common pathways that are affected, but uncommonly you don't see the same exact gene affected by these mutations. So it's still kind of a mystery as to why these are being recurrently altered. But I think by looking at these global changes in the RNA-seq data, we are gaining more information as to what their effects are. So one way that we're looking at somatic mutations that disrupt RNA splicing regulation is through these mutations in splicing factors. And another way that we're looking at mutations in the cancer genomes that affect splicing is through mutations in splicing regulatory elements, or cis elements. So these are mutations either directly at the splice sites or potentially within the exon or within the flanking intron. And all these sequences are known to typically act as binding sites for transacting factors, and these sequences can also be mutated and then affect splicing downstream. So one of the splicing changes that we've characterized previously is in a receptor tyrosine kinase called MET. So this is just a diagram of many receptor tyrosine kinases. These are genes that are frequently mutated in cancer, either through missense mutation, fusion, amplification, and so forth. And so we're looking into also those cases where they're affected by splicing. So it had been previously known, so back in 2006 and 2009, it had been shown that MET is altered through splice site mutations. So here I'm showing particular positions that were shown to be mutated at the 5' splice site, and also these deletions were identified around MET exon 14. And the consequence of these mutations were skipping of exon 14. So in a wild-type sample with no mutation, exon 14 would then, at the protein level, contain a binding site for Sybil, which is a ubiquitin ligase. And so normally, then MET can be ubiquitinated and then further degraded. However, if you skip exon 14, which can come about from any of these splice site mutations, then you would skip the exon 14 and remove that binding site, and so you get a more stabilized protein. And so this was really an interesting event, because typically in cancer genome studies, when you look at splice site mutations, it's always assumed to be loss of function. But this is one case where these splice site mutations are actually causing a gain of function activation of an oncogene. And so as part of the TCGA lung adenocarcinoma working group, we wanted to further characterize this event. And again, we had whole exome and RNA-seq from the exact same tumor types. And from that analysis, we actually identified an unexpected nonsense mutation that was associated with exon 14 skipping. So I'm down here, I'm showing just a model of exons 13, 14, and 15. And then above here, I'm showing coverage of RNA-seq reads in two different patient samples. The top row is a wild-type sample. The bottom row is this one patient that had this Y1003 stop or nonsense mutation. What's interesting is this Y1003 mutation actually falls directly in the exon. And what I'm not showing is that it's predicted to cause a loss of splicing enhancers, and this is something that we're interested in further testing. And so really the take-home message was if we didn't have the matched exome and RNA-seq data, we wouldn't have been able to identify this association. And most importantly, this is a nonsense mutation that's then being is shown to be associated with a gain-of-function alteration. And this is really important in the context of treatment. So what's very promising is that patients that have these MET splicing alterations actually show response to targeted MET therapies. And so by identifying more of these, or understanding all these different ways that MET and other targetable genes are altered, we can potentially help to improve our understanding of how we can target patients for specific therapies. So just briefly, I'll talk about some other goals of identifying additional cis-acting mutations. So we're kind of systematically looking for cis mutations in the exome and whole genome sequencing data, and then asking, do you see an associated splicing change? And one interesting case we've observed is actually in ERB2, which is another one of these receptor tyrosine kinases. So this is out of the 230 lung adenocarcinomas that we originally studied. What I'm showing here is a graph of the expression of exon 16. And there's this one patient, so if you notice, pretty much all patients have about 100% spliced in, meaning most of them are expressing that exon. But there's this one patient that shows a dramatic skipping of that exon, and this was the only sample that actually showed a splice site mutation in ERB2. And just to show that this wouldn't have been identified if we had simply used the annotation, as this is in the transcript annotations, this exon is constitutive. And so N of 1 is kind of risky to make any conclusions on, but what was really promising is sort of just doing some Google searching. We found that this splicing event had actually been functionally characterized before, and so we're further testing this as well. But essentially, in this transformation assay, expressing control or wild type or a known activating ERB2 point mutation versus this exon 16 deletion, what this group found was that exon 16 deletion can also transform very similarly to a known activating ERB2 mutation. And so far, no one, this isoform was reported in breast cancer, it had never been reported in lung cancer, and no somatic mutation had ever been linked to that exon. And again, ERB2 is one targetable cancer alteration, and so we're interested in following up on this. And then finally, we are also involved in the pan-cancer analysis of whole genomes group, and mainly because we're interested in looking for mutations that are farther into the introns, which are typically missed by whole exome sequencing studies. So one example is right adjacent to the exon, there's these splice site motifs, and in typical exome sequencing, we only call mutations right near the exon, but there's additional information that flanks that region, and also it's known that intronic regulatory elements exist, and so we're looking in these whole genome sequencing data to look for additional cases of splicing mutations. And so we'll continue to mine cancer genome and transcriptome data to look at the various ways that splicing can be altered. And I'll end with just thanking my collaborators, particularly from Matthew Meyerson's lab and Kathy Wu for their contributions to the work that I presented. So I'll answer any questions.
Great. We have a few minutes for questions.
So in the big picture of cancer, the splicing mutations are not very frequent. So do they occur in combination with other mutations? Can they drive cancer by themselves? Do you have this bigger picture of their kind of role by themselves?
Yeah, great question. So they frequently do co-occur with known drivers. So for example, in lung adenocarcinoma, the U2AF1 mutations frequently occur with KRAS, EGFR, those kind of canonical driver mutations. When people have tried to make mouse models, introducing the mutations themselves, there's subtle phenotypes, but they don't, you don't see the very strong proliferative effect that you would see with canonical driver mutations. And so people are looking at combinations of mutations together to see what is potentially the additional selective advantage that the factors are doing.
Can you say something about the splicing preference that changed due to the mutation?
Yeah, I didn't. I took that out. So the U2AF1, it's actually really interesting. Well, I guess this is my bonus slide. So for U2AF1, we do actually see different 3' splice site sequence recognition. And so this is the splice site motif of canonical human splice sites. When we see skipping of 3' splice sites, the splice site that's avoided has TAG, and then when you look at it the other way, when you're preferring a more proximal splice site, again you're avoiding splicing at a TAG. So that's one feature of U2AF1. SF3B1, totally different. So you don't see any 3' splice site recognition alteration. What you're seeing, there's some debate, but essentially a differential branch point usage that corresponds to a different 3' splice site change.
Did you try to model it in a way that you can, given the mutation, you can predict?
Yeah, so for U2AF1, I've done that a little bit, and it does seem that there's a strong enrichment that if there's two splice sites that are nearby, and one is a TAG and one is a CAG, there's a majority of those are affected by the mutation. But we haven't gone further to actually build like a stronger model for that. But we do see that when you have those two choices, that there's a strong bias for those to be affected.
I have one question. If splicing factors have these pleiotropic effects, then why don't we see more splicing factors with frequent mutations? Why only these two?
So those were the only two I talked about, but there are actually many others. There's so in lung cancer, there's RBM10. There's been studies on SRSF1. There's also overexpression. There's mutations in SRSF2. There have been a lot that have been identified. There's also other sort of epigenetic modifiers that potentially could also affect splicing, and that has been shown as well. So perhaps they're mutated more frequently, but we haven't maybe identified the causal link. Yeah.
Good. Well, let's thank Angela once again.